Now, I want to talk about the digestion of feed nutrients, which includes the three that we mentioned. We're going to talk about the digestion of protein, starch, and cellulose as they pass down the digestive tract. We'll begin with protein. Digestion of protein that might be in soybean meal, alfalfa, corn, and any other feedstuff that's other than NPN. This would be the protein, where the protein would be. The NPN is separate, and that's the urea. Okay, digestion of all of these in the rumen reticulum involves a breakdown of these to amino acids by the microorganisms to ammonia and carbon skeletons. The non-protein nitrogen breakdown produces ammonia and carbon. Ammonia and the carbon fractions are recombined by the microorganisms into amino acids. They take a product, the microbes take a product that's consumed by the sheep into the mouth. They break it all the way down and then rework it and put it in some order that fits their bodies. They take it into their bodies, then these organisms go down the digestive tract where they are digested into amino acids and are absorbed into the blood and used by the animal. But some of these feed proteins escape degradation by the rumen reticulum microbes. It's called bypass protein. And therefore, the omasum and the abomasum are presented with all of these: feed protein and microbial protein. Enzymatic digestion of these begins in the abomasum and continues, as we said, in the duodenum and jejunum. What are the end products of all of this? Amino acids from the protein are absorbed into the blood and circulate through the body. They're absorbed in the back end of the duodenum and the top portion of the ileum, portion of the small intestine. Starch digestion occurs mostly in the rumen reticulum, unless there's an overload of starch. An overload of starch would be high concentrate diets that might contain 80 to 100% corn, milo, oats, barley, or wheat. That can be an overload as far as digestion in the rumen. Not that anything's going to happen to the animals that is bad. It's just that the rumen will not be able to break all of that starch down and before it leaves the rumen going into the omasum. Starch that's digested in the rumen reticulum yields volatile fatty acids. The main one of these is propionic, and it is used more efficiently than acetic acid. Therefore, concentrates are used more efficiently than are roughages for production. The volatile fatty acids are absorbed, as we see here, through the rumen reticulum and omasum walls into the blood, travel to the liver, where they're converted to glucose. Goes to glucose. Glucose in the blood is used for body cells, for energy, for muscle cell growth, and for fat deposition. Glucose is the end products that's used by the cells of the sheep, just like it is for the non-ruminants. Okay? But it goes a different route than it does for non-ruminant. The starch or sugars go through the volatile fatty acids, whereas non-ruminants do not have the 4-compartmented stomach, so any starch or sugar that we consume goes directly to the omasum, then to the small intestine, where it is broken down into glucose and absorbed. So some feed starch can reach the abomasum undigested if there's an overload. If that's the case, then the hydrochloric acid that is produced in the abomasum begins its hydrolysis and then it is acted on as it passes into the duodenum to yield glucose. And you see where the glucose goes. It can go into fat, it can go into muscle, it can go into to maintenance. So in the ruminant animal, the starch or sugars that they consume and feedstuffs goes to volatile fatty acids, then it is converted to glucose in the liver, and then the glucose goes to the different portions of the body. Cellulose digestion is along the same line, but in the end, it comes out the same. The rumen reticulum receives the cellulose. The microorganisms that live in that rumen we've talked so much about break the cellulose down to volatile fatty acids. These feedstuffs would be something like grass. It could be hay, corn stalks, silage, and you can name a lot of other different feeds. They're broken down primarily to acetic acid. Remember, starch was broken into propionic acid. Acetic acid is not utilized as efficiently as is propionic acid. Therefore, the amount of feed that it takes to produce a pound of gain is less for when you feed a high concentrate ration than it is when you feed a high roughage ration, because the concentrate produces propionic acid from starch and the roughage produces acetic acid, as we see here. Acetic acid is not used as efficiently. Further use of the volatile fatty acids, though, is the same as we talked about with starch. In contrast, that cellulose that reaches the abomasum and the small intestine cannot be digested. If it was not digested in the rumen reticulum, it will pass out in the feces undigested, because there are no enzymes. There are no cellulase enzymes in the abomasum or small intestine that can break cellulose down into smaller units, into volatile fatty acids. Although there might be a little bit of cellulose digestion, as we said early on, that might occur in the large intestine by the microorganisms that live back there, but it's probably minimal. So the total gastrointestinal digestion of cellulose might only be 40 to 60%, whereas we talked about 80 to 100% when we're talking about starch or sugar digestion.